Let
Boa
me do some.
noite, gente. Queria agradecer, né, todo mundo que está aí, tarde da noite, para fazer a nossa apresentação aqui do professor Christopher. Só para poder contextualizar vocês, né, apresentar um pouquinho o professor Christopher. Ele é o professor Christophe Darnault, ele é professor associado da Escola de Engenharia Civil Ambiental e Ciências da Terra da Clemson University. Ele também é presidente da seção da Carolina do Sul da Sociedade Americana de Engenheiros Agrícolas e Biológicos, e ele é um dos representantes da Clemson no consórcio de universidades para o avanço da ciência hidrológica. Ele também foi pesquisador visitante na Universidade de Yale, tem mestrado e doutorado em Engenharia Ambiental e Recursos Hídricos pela Cornell University e possui graduação em Engenharia Agrícola, Ambiental e Biológica pelo Instituto Superior de Agricultura da França. Ele vai falar hoje para a gente um pouquinho sobre o comportamento de agentes e materiais químicos, biológicos, radiológicos, nucleares e explosivos no meio ambiente, como fazer a caracterização do processo de transportes e desenvolvimento de métodos e ferramentas de monitoramento. O professor Christophe, vai falar em inglês para a gente, né? Então eu gostaria de pedir para vocês a compreensão para isso também. Qualquer dúvidas que vocês tiverem, né, ao longo dessa palestra, vocês podem colocar no chat, podem colocar em português mesmo, que no final eu faço a tradução para ele, e se precisar também faço a tradução da resposta dele. E espero que vocês aproveitem muito, né, porque é uma oportunidade muito única da gente ter um palestrante de fora também, né, conversando com a gente sobre um tema tão novo. E é isso, né, espero que a gente consiga aproveitar muito essa nossa noite. Christophe, I would like to welcome you a lot for our seminar. It's such an honor for us to have you here with your expertise in this such new and interesting theme that you are going to present to us. And the floor is yours. Please feel free to start your presentation.
All right. Well, first of all, I would like to thank Professor and Dr. Marina Batalini de Macedo and also Professor and Dr. Ana Paula Moni Silva to give me the opportunity to present to you my research. It's a very great honor for me to share our research done in my research group. So today my talk is on the behavior of chemical, biological, radiological, nuclear, and explosive CBRNE agents / materials in the environment. And we're looking at characterization of transport processes and development of monitoring methods and tools. And I would like also to acknowledge my co-author, Masa Gorban. So Marina introduced myself, so I will also introduce my students. Martha Gorbani is one of my PhD students at Princeton University, doing a PhD in environmental engineering and earth science. She also has a MS degree in environmental engineering at Amir Kabir University of Technology in Iran, and also a BS in civil engineering from Shiraz University. So briefly, my teaching and research interests deal with academic sciences, agricultural and biological engineering, biology, environmental health and engineering, environmental sciences, and also hydrological science and water resource engineering. My research, in my research group, we focus on the soil, water, food, and energy system. We look at environment, resources, and sustainability issues. We focus also on public health, pathogenic agents, and infectious disease. And we also design and developed visualization method tools and sensor systems. So more specifically, we look at the hydrologic, geochemical, and microbiological processes at any random scale that impact both the water quality and quantity in order to protect the ecosystem and the public health. We also look at the influence of physical, chemical, and biological processes on the physics of single-phase and multi-phase fluid porous media with application to agriculture, environment, and energy. We look at the failure and transfer of contaminant into the soil, water, plant systems. We look at the exposure and toxicity of chemical, biological, and radiological agents to public health and ecosystems, and the evaluation of risk posed to human health by contaminations. And also we look at understanding of soil erosions and the key drivers to soil availability dynamics in different soil climates and management practices, and link dynamic soil availability to soil loss and agricultural landscape. So in a nutshell, we look at flow in the surface and the subsurface systems. We look at the contaminant transport into the environment or the soil water plant systems. And as we want to understand the mechanisms, we also have to develop some tools and visualization methods and sensors to characterize and monitor these chemical, biological, and radiological agents to the environment, but also to characterize the environmental systems and processes in the evaluation of risk posed to human health by contaminations. And overall, we look at the soil and water resource management. So this gives you a little diagram of the thing involved in our research. we deal with, of course, human, agriculture, and environment at the center of the work. We work with soil and subsurface systems, looking at flow and transfer and porous media, looking at fair and transfer of contaminant, and soil erosion, which are two big part of our research. For the food side of things, we look at soil health, irrigation practices, fertilizer, crops yield and quality, but also forest issues. On the energy side, we work with nuclear materials and energy, and also fossil fuel. And on the water side, we work with water quality, quantity, water treatment, water reuse, water contamination, public health, and environmental health. So today, our talk is going to be focusing on the chemical, biological, radiological, nuclear, and explosive materials in the environment. I will briefly outline valozone hydrological processes and discuss some of the materials we'll be using. And then we'll move to the research part of things, where we basically will be looking at the chemical side of things, where we look at the feed and transport of nanoparticles into soil, water, plant systems, and also the feed and transfer of nitrate. These are the chemical materials. On the biological side of things, we'll look at the transport of zoonotic pathogens. We work with Cryptosporidium parvoocys and Toxoplasma gondii in soil. And we'll be also looking at nuclear and radiological materials behaving into the environment and the impact of chemicals, but also biochemical compounds on their transport in that zone. And then we'll be dealing with some explosive behavior, dealing with some behavior of explosive materials into soil exposed to prescribed fires. So background, valozone processes and professional flow. So the valozone is the zone which is below the soil surface and above the groundwater table, right? It is characterized with unsaturated systems. So you have water, air into the pores. And it is the locations of bypass transport phenomena called preferential flow, which concentrate the flow into some channels. And therefore, as your water is bypassing most of your soil matrices into the valozone, then you have movement of water, which is non-uniforms. And also with that, the transfer of contaminants, which can be very fast, and therefore bypassing most of the chemical biological reaction that can take place to attenuate these systems. So there are two major types of the preferential flow. One of them is called macropore flow. So here on this system here, you have the blue dye applied to the soil surface, and then we excavate, or the soil was excavated. And you can trace the flow patterns by looking at the path of the dye. And this is an undisturbed soil column or undisturbed soil systems. So the water goes through the roots, crack, wormhole, etc. And that's called macropore flow. Then you can also have some two-dimensional chambers, two-dimensional chamber filled with sand, for example. And then with some light transmission method, you can visualize the flow patterns of the water infiltrating sand or quartz silica sand. And here what is interesting to see is, even though you have a uniform wetting front at the very top, then the water breaks down into fingering, and these are the fingers of the hands, okay, fingering flow. And this is again to characterize the non-uniform distribution of your water patterns. And again, fast movement of your water goes also with facilitate also the transport of the contaminants in it. And you can see here the fingering occurring, right? This is water infiltrating. The black area are just dry sand. So, and that of course impact the water quality. Now, in terms of the work we deal with nanoparticles and biocolloids. And if you look at a triangle that represents the porous media or the pores, you have several locations to retain the particle. In this case here, we have water, right, and air into the pores. And you have, in this case, three major locations. You have here, this is the solid phase, right? So this is to simulate here, you have the grain. This is the pore. So the first retention will be solid water interface retentions. You also have straining over here, number 3, where the particle is being stuck. So here the particle is sorbed to the surface of the sand. And then you have another phase or another locations. It's at the gas or the air-water interface too. And in this case, this is when you have a system which is unsaturated. If you have a saturated system where you have only water into the pores, then you don't have these phases here of gas-water interface. So you have only 2 retentions, solid water interface and then straining. And then you have some small variation of these. We work with many types of nanoparticles, and here we'll be talking about 2 of them. Quantum dots over here. They fluoresce, so they are used for many imaging work, also for electronic display and lighting and other things. And the issue here is the center of the core is made of some heavy metals, so that can be an issue afterwards. And then we also use silver oxide nanoparticles. These are used for their UV radiation substrate capacity. They can be also used into the exhaust gas catalyst in digital fuel additive and many other applications. And once they find the way into the environment, they can contaminate the air, the soil, the water, and the sediment, right? Either after they are being used and put into a wasteland, or following the application into what they're being used for, they can find a way to the environment. So we want to find out what's happened there. Nitrate, so this is a site at Penn State, and Penn State recycles all their wastewater from the campus, from the university, and they apply it to some farmland and forest land. It's called the living filter. And this is in the result of an issue with nitrate released into the streams in the 60s. So now they use that application of irrigations of the wastewater to the farmland, and they want to understand whether or not there's some nitrate issue these days. Biological components, we work with Cryptosporium parvum. So Cryptosporium parvum is released into the environment, has an oocyst of 46 micrometer diameter. In the feces of animals, it is resistant to regular chlorinations. And in 1994, you got 403,000 people who got sick in Milwaukee, and people with young age or elderly people or people with low immunodeficient systems, unfortunately, can be exposed to serious disease and die. So we want to see the impact of the environment on their transport in soil and contamination of subsurface water. Toxoplasma gondii is another type of protozoa that we work with. In this case, it is an issue, especially for women who are pregnant, can induce some problem for the babies. But it also has some other issue with over disease related to brain and also eye issues. But the main thing here is the same thing that for Cryptosporium parvum oocyst. Toxoplasma gondii is resistant to the environment and is resistant to regular chlorinations or regular water treatment. However, the main concern here, I would say, or the main issue about Toxoplasma gondii, it has to go back to a feline. It has to go back to a cat, to a seal, to find its full development, right? So this is the final host for the oocyst to be released into the feces of the cat. But once an animal eats that, it will then be ingested and then can develop some issue with tissue cysts. And then if you eat that, you have also some other issues. So it's a parasitic disease, toxoplasmosis, can give you some significant, again, brain damage and congenital birth defect issues. In terms of the work with nuclear materials and radiological materials, we work with uranium, and in this case, in the Hanford site, these tank who contain some waste of materials which are nuclear and radiological, they leaked, and we want to understand how the dialysis processes and the biological and chemical characteristics of the subsurface will impact the transport of these chemicals into the subsurface, as they have been detected into the Columbia rivers and in other things. And then finally, the final materials we'll be working on is the 1,3,5-trinitrobenzene. This is a highly explosive compound, more explosive than TNT. And we want to see how the soil will react to substrate capacity when the soil is exposed to some prescribed fires. So why prescribed fires? Well, because prescribed fires are the one that we can control, but however, you can see that the whole globe is subject to wildfire and overburning fires, and the change of the substrate capacity of the hydric properties of soil is definitely an issue that needs to be addressed. So let's look at the research that we have been carrying on. So first one is chemical in the environment, looking at nanoparticles and nutrients. Again, we'll be working with quantum dots and serum oxide, and we want to see how these nanoparticles behave into the soil, water, plant systems. So one of the experiments we have done is to have serum oxide exposed to different type of pH, ionic strength, use of fulvic and humic acid. And we want to see how stable is the nanoparticles. So depending on the type of pH, you have aggregation phenomena that takes place. So nanoparticles will aggregate together and form larger compounds. The same thing goes with ionic strength. So the influence of ionic strength and pH have been demonstrated. If you are close to the pH of zero charge, the particle will tend to aggregate. The increase of the ionic strength and the valency of ions, such as calcium compared to sodium, is also demonstrated. And then the use of organic compounds like humic acid and fulvic acid also prevent the aggregation to some extent. And therefore, in this case, if you have limited aggregations, then you can have issue of these nanoparticles being transported. And this is the work again with calcium. So here we have a larger aggregation phenomena that takes place when you have increase of valency. We did, and this is again to see how these nanoparticles remain stable or not, or aggregate into solution, or when they are suspended. We work with transport of quantum dots into unstable porous media. So here we have some silica sand columns, and then we would be applying quantum dots at the top and collecting the effluent. And that system is unsaturated. In this case, we're going to be exploring the impact of ionic strength of the solution and the presence of surfactant. So surfactant here has two main impact. Surfactant impact the flow patterns, regardless of the presence of nanoparticles, right? If you decrease the surface tension, you increase infiltration process or the wettability of the systems. But also surfactant impact the surface-surface interactions. And therefore, it will impact the aggregation phenomena of nanoparticles. So two particles together may or may not aggregate in the presence of surfactant. But also the surfactant will impact the sorption capacity of the porous media of nanoparticles. We tested the stability of the systems to demonstrate that as we increase the ionic strength and the presence of surfactant, we increase the diameter of the aggregate. So increase of ionic strength means that you have aggregation phenomena that takes place. But if you add some surfactant, that
Ô, gente, obrigada aí por ter me avisado que tava sem som. Eu só falei que a gente teve alguns probleminhas aí com a transmissão, mas o professor Christopher já tá voltando. Eu queria pedir só para vocês aguardarem um minutinho que ele já volta. Hi, Christophe.
Hi.
I'm sorry. I think the meeting has ended because of problems here from my part.
Yeah.
But we can we can go back. Sorry for this.
Okay. So am I back in now? Or
Yes, you are back in.
okay. So okay. Sorry about that. All right. So here we have we're gonna start with the figure on the right, and in this case, it is unsaturated systems. So we have the concentration of nanoparticles coming out into the effluent. That's your C concentrations over the initial concentration C0, and that's called a breakthrough curve. And what we can observe here is that we have a decrease of the peak as we increase the concentration of salt, and also we have a delay of the occurrence of the peak. Now, when we add some surfactant, these phenomena have been decreased. Now, if we look at saturated systems where you have no air, right, just the water filling the pores, we also have the same trend where the increase of ionic strength decrease the transport of the nanoparticles. However, if we compare the breakthrough curve peak values, let's say the 0.5 molar here is at about 0.9 versus the 0.5 here is about 0.4. What is the difference here? Same solution chemistry, same materials. This one is unsaturated, it has air in it. This one is saturated, has no air. This demonstrates that under conditions which are favorable to retentions, the gas-oil interface is the primary locations of your retentions and sorptions of materials, compared to a system where you have no air. And the same thing goes with the superficial flow. The increase of breakthrough curve timing, I would say, and also peak is connected to the figuring flow phenomena. So under chemical transport conditions which are favorable, superficial flow / figuring flow allow the rapid transport of your nanoparticles. When the chemical transport condition is unfavorable, where you have the high ionic strength, then the gas-oil interface limit the transport. Now we did the same experiments, but now we're going to be sequentially injecting nanoparticles with increased ionic strength into the same columns without cleaning it. And we see that as we increase ionic strength, we decrease the transport of your nanoparticles. Here you have a peak at 0.05, let's say. However, the 0.05 peak here, right? This is about 0.05. For a molar concentration of 0.5 molar is way lower than this peak here, which is also 0.5 molar, right? But it gives you a peak value of 0.4. So what is the difference between these experiments and this one? This is single column use, right? And this is sequential injections into the same columns. So we demonstrate that which demonstrate that as you inject nanoparticles into the systems, some of them are retained, and that's known as retention phenomena. But the presence of already retained materials facilitate the retention of others, and that's called the rapiding phenomena. Now, if you add surfactant, that phenomena tend to disappear, of course. Then we did some work with nanoparticles into cellular proximity, looking at different type of chemistry. Again, the pH, the presence of citrate, organic matter, and we use ICP-MS to quantify the amount of nanoparticles into the effluent and into the sand systems. And what we can see here is the effect of pH. So getting close to the point of zero charge, you have aggregations, aggregation phenomena, and retentions. And that's with the breakthrough curve here on the left figures. And then you can connect that with the retention of your nanoparticles into the sand. So more is at the surface or in the top part due to filtration theory. As you go downwards into the outlet, you have less transport and less retention. Again, if we now add citrate, some of these chemistry changes, and you can either enhance the transport or here the pH 1.5 has demonstrated an increase of the transport compared to the previous one where you had immediate transport. Same thing with organic matter and the use of the nanoparticles. In this case, most of your nanoparticles are being transported easily into the soil. And then we model the breakthrough curve of the transport of the nanoparticles under these different type of conditions. So we have DI water with pH 1.57 and the use of different compounds like citrate and organic matter. And we're able to simulate the transport of these nanoparticles into the subsurface. We did the same thing with the retention of nanoparticles and the retention profiles with some modeling of these nanoparticles into the subsurface. So if we now compare our data in the symbols versus the different type of modeling that we use, we are able to quite well model the transport of the nanoparticles into the effluent. However, here, if you look at the modeling result of the retention modeling, it doesn't do good. So we're not able to match the retention profiles with the modeling. And this can be due to the fact that you have different sizes of nanoparticles and the models only use one size. So aggregation phenomena is dynamic, you have different size, and therefore we're not able to catch that. Also, you have some attachment and detachments of your system that also can create that. So there's different options for demonstrating that the retentions modeling was not as good as it could be. We also like to see things. So in this case, we're going to be visualizing the sorptions and straining, so which are the two retention phenomena into saturated flow conditions of quantum dots being exposed to solution chemistry that favor these. And therefore, we use some laser microscopic scanning, which is a Zeiss LSM 710 inverted confocal microscope. So here we have the laser that's going to go from bottom up, and we have a small chamber filled with some sand and a solution chemistry. And we're going to be injecting the solution chemistry on one side and quantifying the effluent on the other side. And at the same time, we're going to be having the laser to scan the pores to visualize the quantum dots. So what we can see here, so this is a sand grain. This is another sand grain, another sand grain, another sand grain, another sand grain. And this is the void. This is the water filling the void of the pores. And these are slices, okay? And you can see two things here. You can see that the nanoparticles aggregate because we have adding strength of 1 molar. So it promotes aggregation. So the aggregates getting larger and larger, so you can easily see them. And then in this case, they are flowing through the pores. And then you can see here a line, and that's the border between the sand grain surface and the water into the pores. And that's to demonstrate the solid water interface retentions, so sorption of the nanoparticles. And then as we move somewhere else into the systems, we can see here an aggregate which is stuck by straining. And here we can visualize both retention of straining and solid water interface or depositions. And then we can do some 3D reconstructions to visualize that. And then as we monitor the effluent, we can see that from 0 to about 7, there's no transport of the nanoparticles into the postmedia because they are being retained by sorptions and retention and the straining, excuse me. I also would like to add that there's also a sedimentation process that took place, okay? So nanoparticle settles also. But as we switch the solution chemistry from 1 molar to DI water, we had the release of your nanoparticles which were retained. And this demonstrates that the phenomena was reversible. So the retentions is reversible, indicating the existence of secondary energy minima where the nanoparticles is retained. So in this case, nanoparticle get stuck or get sorbed, or aggregate, but they can now deaggregate and being removed from the solid surface and being transported again. So which is probably important because when you have change of social chemistry, that's an issue. So now we're going to work with plants and roots and trait. So the plant trait is like the plant compositions and the roots are the roots. And we want to see the impact of nanoparticles don't have calcium oxide on the root development and the plant trait of sorghum. So that's for the root plants. We have different type of species. We have the sorghum gracile BTX623 Rio, and we have increased nanoparticles concentration in soil from 0, 100, 500, 1,000 milligram per kilogram. And this is what we did. After 3 or 4 weeks, we collected the roots. This one here is not up to scale, but that's to show a picture of how the process works. And what we've done is we're going to be scanning the root, and then using a software, we're going to be able to isolate the primary and the lateral or the secondary roots. And then we can quantify their size, volume, etc. And we did that for all 3 types of sorghum. And this is some of the results. So we can see that for the primary root length, in these cases here, it was BTX623 and the gracile, which after 500 milligram per kilogram were subject to some impact. However, Rio was not. These show 2 things. Number one, that yes, nanoparticle has an impact after a threshold on the development of the roots, but also it is species specific because not all of them will be impacted. Rio was not. Same thing goes with the numbers of lateral roots. We can see some trends where after some threshold you have an impact, and that also can be gradual with the change of concentrations. The plant trait, so we grow the sorghum into a greenhouse, and then we harvest, we process the stem, we process the leaves, and then using some near-infrared spectroscopy, we can quantify different type of materials, about 70 materials or 70 compositions or components of the sorghum. We also monitor the accumulation of nanoparticles into the plant. As you increase the concentration of nanoparticles into the soil, you have the increase of the uptake, and ultimately, of course, the leaves. So this is S is for the stem and L is for the leaves. You have the increase of concentration of nanoparticles into the stem and the leaves, but with the leaves being the ultimate locations. And then as we obtain many type of results for the different type of parameters where we're looking at, we look at bioenergy, materials, we look at bioenergy parameters. We also look at nutrition parameters and others. And basically, there are different observations. In some cases, the nanoparticles increased concentration has no impact whatsoever on the trait. Okay. In other cases, you can have a threshold effect where after some level, yes or no, you have the increase or decrease of the compounds based on the limit. In other cases, you can have some increases as you increase the concentrations of your nanoparticles, okay, or a decrease. So as I mentioned, we work with Penn State and to look at nitrate transport in karstic systems. So karst are big caves or big structures that evolve over the years in geological formations. And they are characteristics of the subsurface of the Penn State of some part of Pennsylvania. So as I mentioned, they recycle all their wastewater year-round for irrigation of farmland and forest land. And we want to see the nitrate transport and the concentrations throughout the systems. So this is the site locations of the leading filter. And we work with this site over here, which is the astronomy site. And over here, this is the farmland. And over here, this is the forest. And you have some depressions that are observed, and some fractal mapping can be detected. So that's also to demonstrate the connection between surface and subsurface systems And the hydrological properties of the systems are characterized by high hydraulic conductivity, and these are some of the subsurface systems. So you see this big cave or big thrust fault that are present into the subsurface, and water can go very fast there. Also, the underdrain valley of the big hollow. So you have the karstic terrain, right? So karst, big conduit in the subsurface. So what we've done is we model the watershed and the subsurface using mud flow first, and then we compared that to some already published work or work available. And once we have the good sense that our model was appropriate for groundwater flow into karstic terrain, then we use MT3DMS to be able to quantify the nitrate into the subsurface for the entire systems. And I would like to remind you that's dealing with karstic terrain. And then we were able to compare the data of 2015 to some observed data. And once we were comfortable with the model, we were able to predict the impact of different changes of application of wastewater to the system for 2035. And in all cases, we're able to meet the nitrate criteria. Work with biological compounds, pathogen, oocysts, and Toxoplasma gondii. So in this case here, we'll be looking at some analysis of your compounds into water and soil, and we can either use microbiological analysis or DNA analysis. So in this case here, we use some visualization inspections using some immunofluorescence staining. In this case, we're going to be looking at the second row here. The staining demonstrates that you can identify a oocyst and the fact that it is a green dye on its wall. And in this case here, if you have the inside is red due to another chemical, this one is dead, and this one is alive. Okay. Nowadays, we use DNA analysis and real-time polymerase chain reaction to quantify the oocyst into the soil or into the water. So looking at different type of phenomena here, we'll be looking at the transport of the pathogens, the oocyst, into stand columns. In this case, we apply feces from the calf, and then we collect the effluent in the systems. The system is made of removable rings. On the right, you have a slice and a picture. So this is dark, means you have water, and that's unsaturated flow like the fingers. And and this one is dry. Due to the physics, the bottom is saturated, okay? But most of the column is unsaturated. And then we have the tracer also applied as we apply the pulse of the pathogens. And we can see two things, that the peak of the pathogens, the egg of the particles, is earlier than the peak of the solute, which makes sense because they are less subject to diffusions. And that illustration here demonstrates the fingering flow and high concentrations of your pathogen to the effluent water. Now, as we slice the columns, we can see the water saturation profile, saturated at the bottom, less saturation as you go up into the column. And then we can determine the amount of pathogens at different levels. What we looked at was that at the bottom here, we had some spikes, and the spikes here connected to this area here. This area here is called the capillary fringe. Over here, this is saturated, and then it is decreasing its degree of saturations. Over here, the capillary fringe is the zone of high amount of air and water compared to here where you have high amount of air. And this presence of gas or interface can lead to retention of nanoparticles, can lead to retention of the pathogen, the particles, and its increase. So to demonstrate that, we artificially created two zones of water repellent sand. So we have one zone over here and another zone over here, which means that if the sand is water repellent, just this zone, then above that, water will not penetrate. And therefore, you have the increase of your water content and the increase over here. And A and B are to replicate. And with that, with these artificial creations of the gas-oil interface, right? Then we want to demonstrate that we have connections of the peak or connections of the increase of retention of your pathogens, as demonstrated here. This peak and this peak connect with these two peaks, right? So under the same conditions of flow and systems, we have the increase of retentions, right, into the soil, because if you look at the breakthrough curve here, you have less than 0.1, and before you had like a 0.3. Okay? So that's to demonstrate the role of gas-oil interface. We did some modeling to be able to, predict the behavior of the pathogen to the subsurface. We did further analysis with soil, real soil, and the impact of surfactant. Why surfactant? Because they are used everywhere for soil conditioning, wastewater applications, and other type of pesticides, so they're fine to the environment. And we choose aerosol 22 because it was showing some different surface tensions compared to the soil we tested. And here are the experimental apparatus. So we have the soil columns, we have some different rings where we can remove we can slice the column at the end, so we can look at the distribution of your pathogen to the soil and also collect the effluent. And we will be studying the impact of surfactant on the transport of your pathogens. Again, surfactant because it impacts the flow patterns and also the interactions of particles with surface. These are some of the pictures after the experiments where we can see the impact of the surfactant on the different type of soil. And what we're going to be observing here is the dots are the pathogens and the lines are the tracer. So basically, we had interesting findings because in some cases, you can have the increase or decrease of the transport in the presence of surfactant. So you would think that surfactant can increase the transport. Well, guess what? In some cases, it did the opposite. And what we observed was also in connection with the retention into the soil profile, where basically we have, in some cases, increased transport when you have the surfactant. There's just no increased transport when you have the surfactant. And this was done in to repack soil columns, so where we have nice sorted soil materials. But we also did some similar testing into undisturbed soil columns, right? Intact soil columns like this one here. And we applied the nanoparticle, we applied the pathogen at the top and collect the effluent. Stem structures of soil, same chemicals of aerosol 22 for the surfactant. So here we're going to be again seeing the same phenomena where in some cases the presence of surfactant. So here I have no surfactant. We're going to be focusing on the first columns and the third column first. Okay? So no surfactant. If I add my surfactant, yes, I have increased transport. I'm happy. That makes sense. But for the Guilford soil type. Same thing with green sun. I have no surfactant. I add some surfactant at 2 CMC, 2 critical missile concentrations. I have the increase of my transport. But now for Lewiston and Sparta, when I have no surfactant, I have transport. If I add surfactant, I have no transport or limited transport. So again, showing the opposite effect. And then if I'm using half CMC of concentrations, I have absolutely no transport whatsoever or no movement of your pathogen into the soil. It's being retained. Same thing goes with your breakthrough curve. We have similar findings for the first 2 rows. No surfactants with 2 CMC surfactants. Now we have the increase of the transport. And then for Lewiston and Sparta, there's no transport at all. And then the same thing goes with half CMC. So we did some modelings with curve fittings. We have, optimization of the model as you increase the numbers of parameters. But what was interesting is as we look at the effluent, we were able to see some flux. And one of the rationale is that the presence of the surfactants introduced some flocculations of your water and reactions with the soil compounds and the chemicals of the surfactants. And these flux, right, they obstruct the pore, reduce the hydroconductivity, and therefore can decrease the transport of the pathogen. So half CMC concentrations created the larger flux and therefore the limited transport. We did the same work with the other pathogens, Toxoplasma gondii, and we had, this is the DNA analysis, and we have similar trends where, in this case, for that particular Toxoplasma gondii pathogens, the presence of surfactants usually increase the transport of your pathogens into the subsurface. So again, now showing that different type of biological compounds will react differently with, of course, the same chemicals and the same soil. And now let's look at the uranium transport. So we work with uranium-6 and uranyl phosphate doped with uranium-235. So again, the issue here is that these tanks are leaking, and we want to see the impact of that is on hydrological processes and chemical and biological compounds on their transport. So here we use unsaturated flow experiments where we have some columns filled with sand, and then we inject the uranium at the top and collected the effluent. And we tested the impact of chemistry, pH 6 versus pH 11. So here we have the tracer, and we have the uranium-6 being monitored. There is a delay, right, of the transport with pH 6 when you compare that to the tracer. So comparisons of your uranium-6 breakthrough behavior at pH 6 with that of the non-reactive tracer indicated that uranium-6 transport was significantly retarded. About 52% of the added uranium-6 absorbed to the cracked sand, likely in the forms of UO2 OH+ and UO2 2+. Now at pH 11, we compare that, and we can see that there is the breakthrough curve of uranium-6, which increased gradually, right, and reached a plateau at the value oscillating between 72 and 82%. And then we also monitor silica into the effluent. So it's silica sand, right? So upon reactions with the pH 11, silica was released from the dissolution of the quartz sand, which allowed the possible transport of uranium-6 following precipitations of uranium-6 containing solid, such as uranide silicate minerals or substance of uranium-6 on silica colloids. And then we can do some mobile, immobile transport and reactive transport of the systems. And again, using some chemical non-equilibrium processes characterized by different type of substrate kinetics, and also physical non-equilibrium processes with some flow regime where you have some mobile and immobile water, we can model the transport behavior of these uranium. We also developed some additional characterizations of the radioactive materials using gamma radiations. So here we have some gamma ray probe. It's in the back here. So that probe can be mounted and can move up and down, left to right. And we have here these two-dimensional tank filled with sand. And we had buried 1, 2, 3, 4 uranium sources. So uranyl phosphate doped with uranium-235 into that systems. And then with the probe behind, gamma ray, we can quantify or detect the uranium systems. And we have the rainfall system here that can apply chemicals and water, and we'll be looking at their impact afterwards. But what was demonstrated here is that we can quantify the uranium into the systems based on the with background. So the energy calibration was at 186 keV for the uranium-235. So we can detect the uranium into the systems by counting times. So that probe here will move up and down, left to right, and can be automatized to stay at any location for some time to count and have some sense of the amount of uranium. And then and this was for uranium, yes, uranium-235. And then we detected also uranium-238 into the effluent. And the fact that you have some citrate, for example, induce the transport compared to other type of chemicals, again, to demonstrate the roles of the plants and also to some extent microbes on the generation of these compounds and the ability to move the uranium into the subsurface. These are some of the capability that we are in our lab. These are initial work where we have here some sensors that react to oxygens and also to pH, right? pH oxygen sensors. So we can also monitor the change of pH and change of oxygen into the systems while imaging the water content, but also imaging the concentration of uranium. And finally, the explosive material into the environment. So the 1,3,5 trillion benzene. So we assess the impact of prescribed burn on the absorption process of a polar and a non-polar compounds. But we find out afterwards, of course, that one of them was a highly explosive compounds. So prescribed burn is used to burn the small bush into a forest to avoid excessive fire, usually, and also for management. But it's definitely going to change the soil properties, and we want to see how these impact the absorption properties of these soil on this type of compounds. So here we had access to a watershed 80 and watershed 77, and another watershed. Here, the watershed AA. But the watershed 80 is the control, so there's no prescribed burn. The watershed 77 is where they have prescribed burns every 2 years, about every 2 years. And here they have the exact same soil. So this is your control site, and this is the watershed 75, same soil here, but prescribed burn and control. And then we characterize the soil. So this is a sandy loam for the watershed control 80 and 77. And then we did some absorption batch experiments where basically we're going to be exposing the different type of compounds with different type of amount of soil for some time, and then seeing how much is being solved versus how much remains in solutions. So these are the information about the chemicals, the 1,3,5-trinitrobenzene, 3-chlorobenzene, and the 1,3,5-trinitrobenzene. So here we have different concentration of soil for the same amount of chemicals being placed. And then we can analyze the TCB through these gas chromatographic equipment with some electron chemistry detectors. And then the TNB compounds is analyzed with some high performance liquid chromatography equipped with some ultraviolet detectors. And what we can see is if we look at top means for top soil, right? So that the soil which is exposed to burn, or that the soil which is not exposed to burn in the case of control. But if you look at the top soil KD value, when you have a prescribed burn, you have an increase of the KD value. Okay? And the bottom KD is less subject to fire because it's the bottom part of the soil, right? So this demonstrates that the burning has an impact on the absorption capacity of the soil, increasing its absorption capacity when the soil is burned for the TCB compound. And the same goes with your TNB or your polar compounds, trinitrobenzene. The top KD is 174, and the top KD absorption capacity for your soil is 285 or 86, so highly increased. Again, demonstrating that the prescribed burn or the presence of fire impact the absorption capacity of soil on these chemicals compounds. And you can see the percent of difference here. So the top 348 to 451 and 174 to 283. It's about 25 and 45% changes. I would like to acknowledge my collaborators and also support provided by USDA and the Department of Energy, and also Thames University. Thank you.
Thank you, Christophe. It was so many new things for us to learn.
Yeah.
You have done so many experimental work and modeling work. It was really amazing to see all of these different compounds that you have explored. I have some questions for you here.
Sure, yeah.
The first one is about, I was actually going to ask if you have done some modeling in watershed scale, and I see that you have made that for nitrogen with the mod flow. But for the others, have you tested also for how does this transport occur in a watershed scale?
No, we have not done that.
Okay, and for the nitrogen, does it account also with plant uptake, this model that you have tested, or is only about transporting in the sediment in the soil?
That's a very good question. I will have to dig in. We published that work, so that should be available. I do not remember on top of my head all the parameters that we use, but it's something standard from the groundwater flow models and the nitrate transport models, and I can share with you that information. We published that work, so. I don't remember.
Yeah,
I don't remember. I don't remember all the nitrate
Yes.
mechanism that were yes or no included.
Okay, I'm asking you that actually because, as you know, I have been working with bioretention experiments in general, and we have done we are trying to construct actually a bioretention model to account with the hydrological part and also with the pollutant treatment. And we have to use some pollutant transport in the soil of the bioretention. And since it's not our specialty, we have been actually having a hard work to do that. So your work, your presentation from today gives me so many inputs in my mind of how can we proceed with that. And I would love to share this with you later so we can find a way to collaborate on that.
That would be great.
And I have a question here that is from students from environmental engineering course that they have asked if you have tried to do some experiments on bioremediation on this some of these components.
No. I have not done, to what I can remember, I have not done any work with bioremediation, to my knowledge. Yeah.
Okay.
We only look at the transport of the contaminant.
Yeah,
we
I
can't
would
do
imagine. And it is so much work.
Other people can do that, but
Okay. Great.
I
will give them the idea to use your transport to analyze if it's possible to do some bioremediation on that.
Yeah.
Okay, so great. I would like to thank you so much for your time to join us in our seminar. It was really enrichment for us to learn a little bit more about this behavioral, all of these compounds that you have analyzed. And I hope we can keep on collaborating with each other. And maybe someday you can visit us here in Brazil.
That would be great. And same thing here too,
So I think that's it. Thank you so much again, Christopher, and I hope we can keep in touch. Thank you.
Okay, well, thank you so much, Marina. I appreciate it. It was an honor for me to present to you and your students and colleagues. Thank you so much.
Thank you.
Bye-bye.
Bye-bye. Gente, a gente vai terminar aqui então a nossa transmissão, agradecendo de novo, né, ao professor Christophe pela presença dele aqui, e espero que vocês tenham aproveitado bastante. Lembrando então, né, que amanhã a gente continua com a nossa programação e espero ver vocês lá presenciais na Unifei, tá bom? Tchau, boa